Okay, so we just looked at the active side a little bit and some issues relating to specificity. So let's keep thinking about that and move on to thinking about substrate binding, substrate activation, and catalysis. And then we'll look at some ribozymes, which are kind of cool and new and interesting for you guys. So if we think about a reaction occurs when two substrates come together, and the reaction will go faster if the two substrates collide with a catalyst. And obviously, in the context of a cell and cell biology, that catalyst would be an organic catalyst, a protein. And so the reaction will go faster if you increase the concentration of the substrates, because therefore there's a greater chance they'll collide with an enzyme, and also if you increase the concentration of the enzyme, because then there's more likelihood of the enzyme colliding with the substrates. We can also increase reaction rates and make them more efficient by putting enzymes and substrates in the same place. We talked about that a little bit. You can also think about not just putting enzymes and substrates in the same place, like in the same kind of specific structure, like a peroxisome with catalase and hydrogen peroxide, for example, but you could also put the enzymes in a membrane, and therefore you restrict their movement from 3 dimensions to 2 dimensions. And so you see this nice example of this. You see photosystems and electron transport chains in photosynthesis being arranged into membranes, and you also see the electron transport chain of the last step in cellular respiration, or prior to ATP synthesis, rather. You see the electron transport chain proteins arrayed in membranes, so that the substrates of the products of one enzyme can be passed to another, and in many cases that's a reduced electron carrier. So there are strategies for increasing enzyme efficiency and the rate of catalysis. And like we talked about last time, the precise chemical fit of a substrate into an active site means that you have a lot of specificity in many, many enzymes. Once the substrate binds into the active site, much like this D-glucose molecule here binding into the active site of hexokinase, if you don't remember, that's the first enzyme in glycolysis, you have a specific binding into the active site, and then once the substrate is oriented correctly, that will facilitate the reaction chemistry. And so the interaction between the substrate and the enzymes, often involving hydrogen bonds, ionic bonds, in some cases both. Rarely there is a covalent bond formed between the substrate and the and the active site. Now, some of you will have been taught about what's called the lock and key hypothesis, and this is actually kind of a little bit odd, because this was really what we believed kind of in the 1890s. Here's Henry. This is the big one, Henry. And that's where we believed that the active site was rigid and the substrate bound rigidly into the active site, but that's not really kind of how it works. It's more accurate to think about what's called induced fit, that when the substrate binds into the active site and in some cases a lot of the other structures in the protein change shape to better accommodate and facilitate catalysis in the active site. So rather than thinking about a lock and a key mechanism, I think the best way to think about this is if you take a softball mitt or a baseball glove, and when you put your fingers into a baseball glove or a softball mitt, and you kind of get them in there, and you can probably catch and toss, but it's not very efficient. And so you kind of wriggle your fingers around a little bit, and then you kind of really get it in there. And the whole structure of the mitt changes. It often folds your fingers over. And so that's kind of how I've always thought about induced fit, and maybe that helps a little bit. So the conformational change that occurs brings kind of essential and needed amino acids and their side chains, their R groups, into the active site. It's all about the R groups, obviously. And sometimes those R groups are amino acids that aren't even that close to the active site, and there's a significant shifting of enzyme structure that bring the R groups into the active site, and that holds the active site in a conformation which makes the substrate kind of bound in there much more efficiently, and makes the active site optimal for catalysis, if you like, and that helps really distinguish between non-specific binding into the active site, because some molecules are going to get in there, but they're not going to induce the induced fit structure, and so they're often going to just be released. So there will be things going in and out, and only the things which go in, which are specific for the active site, induce the conformational fit, and that brings in the R groups, which are often going to be involved in catalysis. So over here we've got the unoccupied active site catalytic residues of carboxypeptidase, and this enzyme is involved in breaking down proteins in the GI tract, and also in terms of protein maturation, which we've not really talked about. But sometimes you have to cleave the end of a protein to make it active and so that it folds properly, and that's one of the things that carboxypeptidase A does. And so you see there's a glutamate amino acid 72 here, GLU72. So the 72 refers to the amino acid number in the primary sequence. Histidine 69 and then histidine 196. So you can see these three amino acids are, these two are close together, 3 amino acids apart, and you can see that little loop down here. But then histidine 196 is 124 amino acids away from this glutamate. But they're all being kind of brought together by the three-dimensional structure of the protein, and they're coordinating here a zinc ion, and those could be ionic bonds or hydrogen bonds, and they're coordinating a catalytic zinc. So if the zinc's not coordinated there by these three R groups, then the enzyme's not going to be functional. So think about all the trace nutrients you need, things like zinc in your diet, iron in your diet. They're often involved in catalysis in enzymes. So when the substrate binds in here, and the substrate is a short peptide, and it's kind of shown here in red in the background. Once that's bored into the active site, think about that as just a short piece of peptide from another protein that's going to be metabolized by the carboxypeptidase, and the induced fit is triggered. That causes other R groups to be brought in, the glutamate 270, tyrosine 248, arginine 145, and some of these will be the catalytic residues, and so they will either donate protons or accept electrons, things like that. They'll cause bond distortion and things like that. And you can see that the glycine here is interacting with the zinc, and so without that interaction there, then you wouldn't get the change in the conformation of the protein. And so once you get this induced fit and you've brought these other amino acids into the active site of the enzyme, then you can get more efficient catalysis. So you often get these very specific interactions between the substrate going to the active site and the active site amino acids and the catalytic amino acids as well. And so once you got the substrate into the active site there, often you have activation of the substrate so that it can be completely modified. And so we're seeing that here in the active site of rubisco, which is, if you remember, ribulose bisphosphate carboxylase oxygenase, and that's the enzyme which is involved in adding carbon dioxide, the carbon of carbon dioxide onto ribulose 1,5-bisphosphate, the first step of carbon fixation in the Calvin cycle. And so here we've got RUBP in the active site, and we've got all of these hydrogen bonds which are being made between active site amino acids in the active site and the substrate. And so as the substrate becomes activated, several things are going to happen. So we may see the enzyme kind of twists or distorts the substrate slightly, and that causes some of the bonds in the substrate to be strained, so they're more easily broken or catalytically attacked. We may see proton transfer occurring between active site residues and the substrate, or vice versa. We may see the donation or acceptance of electrons. So if we're certainly going to be dealing with oxidation and reduction reactions, then part of the process is going to be getting the substrate ready to accept or donate electrons. The last thing is that then we would have catalysis, and so and that's going to be the kind of final processing of the substrate, either reduction or oxidation, cleavage, rearrangement of a functional group on the substrate, that kind of thing. Once that's happened and the substrate has been modified into a product, and bear in mind there may be two substrates in the active site, so there'll be two sets of interactions going on. Once the substrate or substrates have been modified, the active site will then change again and release the products, and that resets the enzyme. So you got to think about this happening on the time scale of 10 to the 7 events per second. Thousands and thousands of times a second some of these enzymes work. So the chemistry is pretty remarkable. This is one of the amazing things about biochemistry, is the speed which these things work and how efficient they are at doing some of these things. But don't forget, the whole thing is triggered by the random collision of a substrate or substrates and the enzyme. And so we can go back to thinking about the things which promote that initial interaction. So that's substrate binding, activation, and a little bit about catalysis. The next time we'll talk about ribosomes. No, we won't. We'll talk about ribozymes.